Welcome, everyone. Today, we are diving into the fascinating world of phylum Annelida, commonly known as segmented worms. If you have ever seen an earthworm after a rainstorm, you have already met an annelid. But these creatures are far more complex and important than they appear at first glance. In this lecture, we will build your understanding from the ground up. We will explore their unique body structure, the physics behind how they move without any bones, and how scientists classify them into different groups. Let us get started. Let us begin by picturing something you have likely seen, an earthworm wriggling on the sidewalk. If you look closely, its body is not a smooth tube. Instead, it looks like a stack of tiny, identical rings joined together. This ring-like appearance is the defining hallmark of the annelids. To understand this intuitively, imagine a long freight train made up of dozens of identical boxcars connected in a line. This is very similar to how an annelid's body is built. By dividing the body into these repeated compartments, the animal gains incredible flexibility and precise control over its movements, allowing it to navigate complex environments. Now that we have a visual in mind, let us look at the formal biological definition. Annelids are characterized by two major evolutionary advancements. First, they have a true coelom. This is a fluid-filled body cavity completely lined by tissue derived from the mesoderm, acting as a protective cushion for their internal organs. Second, they exhibit metameric segmentation. This means their body is divided externally by shallow ring-like constrictions called annuli and internally by partitions called septa. These divisions create a series of repeated segments, or metameres, which is the scientific term for those boxcars we just talked about. Building on their segmented structure, we must also understand their embryonic development. Annelids are triploblastic, which simply means their bodies develop from 3 primary embryonic cell layers. Additionally, they exhibit bilateral symmetry. If you were to draw a line down the center of an annelid, the left and right halves of their bodies would be mirror images of each other. Why does this matter? These evolutionary advancements are crucial because they allow for much more complex organ systems. Combined with their segmentation, this allows annelids to perform highly coordinated movements compared to simpler, unsegmented worms. You might be wondering why these specific traits make annelids so important to our world. They are actually crucial to the survival of our planet's ecosystems. Earthworms act as nature's plows. By constantly burrowing, they aerate the soil and enrich it with nutrient-dense waste, which is vital for our agricultural crops. In the medical field, the medicinal leech has been used for centuries. Even today, their saliva is utilized to prevent blood clots during delicate reattachment surgeries. Furthermore, in marine environments, countless species of ocean-dwelling annelids form the foundational base of the food web, feeding fish, crabs, and birds. Understanding their ecological role leads us to a fascinating question. How do they actually move through tough soil without bones? The answer lies in their hydrostatic skeleton. Because of their metameric segmentation, each segment of the worm acts as a closed cylinder filled with coelomic fluid. When the worm's circular muscles contract, they squeeze this fluid. Since fluids cannot be compressed, this squeezing drastically increases the internal pressure. This pressure has nowhere to go but outward against the segment walls, generating a powerful forward force that drives the worm through the dirt. To truly grasp how this hydrostatic skeleton works, we can represent it mathematically. The pushing force of the worm is governed by the equation F equals P times A. Let us break down every symbol. F represents the outward force generated by the worm's segment, measured in newtons. P stands for the internal fluid pressure created by the contracting muscles, measured in pascals. Finally, A represents the cross-sectional area of the worm's segment, measured in square meters. Conceptually, this tells us that the pushing force depends directly on how hard the muscles squeeze and how thick the worm is. If the internal pressure increases, the burrowing force proportionally increases. Now that we have our formula, let us apply it to a real-world scenario. Imagine a large earthworm trying to burrow through compacted garden soil. We are given that the circular muscles create an internal fluid pressure of 2,000 pascals. The cross-sectional area of this segment is 0.00005 square meters. We need to find the total outward force. We choose our hydrostatic force formula because it directly relates pressure and area to force. We substitute 2,000 for P and 0.00005 for A. Multiplying these gives us a final answer of 0.1 newtons. This physical mechanism explains exactly how soft-bodied annelids conquer solid environments. Having calculated the force, let us visually examine the anatomy that makes this possible. On screen is a cross-section of an annelid. Notice the external rings, labeled as metameric segments or annuli. Inside, you can see the vertical partitions called internal septa, which divide the body into those boxcar-like compartments. The blue area represents the coelom, the fluid-filled cavity we discussed. Look at the red arrows pointing outward. These represent the hydrostatic pressure generated when the muscles contract, pushing against the body wall. You can also see the digestive tract running through the center and tiny bristles called setae at the bottom, which help grip the soil. Beyond calculating force, the segmented nature of annelids allows us to easily estimate their physical size. Because the body is made of repeated, nearly identical units, we can find the total length by multiplying the number of segments by the length of a single segment. The formula is T equals N times L. Here, T is the total body length. The variable N represents the total number of segments, or metameres, and L is the average length of one individual segment. If a worm grows by adding more segments, the value of N increases, and therefore the total length T increases, demonstrating perfectly modular growth. Let us see how a marine biologist might use this length formula in the field. Suppose a biologist discovers a fragmented polychaete worm. They count exactly 120 identical segments. Microscopic measurements show each segment has an average length of 2.5 millimeters. We want to find the total estimated body length. We use our modular length formula, substituting 120 for N and 2.5 for L. Multiplying these together gives us 300 millimeters, which equals 30 centimeters. This simple arithmetic is incredibly useful for estimating the true size of marine worms that often break apart when collected. Now that we understand their anatomy and mechanics, we must look at how scientists categorize this massive group of animals. According to the widely accepted classification by Barnes, the phylum Annelida is primarily divided into 3 major classes. This division is like learning the branches of a family tree. The categorization is based largely on 2 physical features: setae, which are tiny bristle-like structures, and parapodia, which are fleshy, paddle-like appendages. By looking at whether a worm has these structures and how they are arranged, we can see how different worms have adapted to completely different environments, from the deep ocean to your backyard garden. The first and largest branch of this family tree is class Polychaeta, which literally translates to many bristles. These are almost entirely marine worms, like the clam worm or the beautiful Christmas tree worm. They are highly developed, characterized by having a distinct head, complete with eyes and tentacles. True to their name, each of their body segments features a pair of fleshy, paddle-like parapodia. These parapodia are covered in numerous setae. The worms use these bristly appendages not just for swimming and crawling along the ocean floor, but also for breathing, as they act very much like gills in the water. Moving from the ocean to the soil, we encounter the second class, Oligochaeta, which translates to few bristles. The most famous member of this class is the common earthworm. Unlike their marine cousins in Polychaeta, oligochaetes do not have a distinct head, and they completely lack those fleshy parapodia. They do have setae, but as the name suggests, they are very short and few in number. These tiny bristles are used primarily as anchors to grip the soil as the worm's hydrostatic skeleton pushes its way through the earth. They're mostly terrestrial and freshwater dwellers, playing a vital role in soil ecology. The third and final major class takes a highly specialized evolutionary path, class Hirudinea, which comprises the leeches. During their evolution, leeches completely lost both the fleshy parapodia and the bristly setae. Instead, they developed powerful suckers at both the anterior and posterior ends of their bodies. They use these suckers to attach to hosts and move in a looping, inchworm-like fashion. While most leeches are found in freshwater and are infamous for feeding on the blood of vertebrates, some are actually active predators that hunt small invertebrates. Their unique adaptations make them distinct from the other two classes. As we wrap up our journey through phylum Annelida, let us review the most critical concepts. First, remember that these creatures are fundamentally defined by their true coelom and their beautifully segmented bodies. Second, this metameric segmentation, combined with a pressurized hydrostatic skeleton, gives them incredible mechanical advantages, allowing them to generate force and move through dense soil without bones. Finally, we learned that they are classified into three main groups: Polychaeta, Oligochaeta, and Hirudinea, based largely on the presence of setae and parapodia. You now have a solid scientific grasp of some of the most ecologically important invertebrates on Earth. Great job!